Hi, my name is Love, and today I'll be going over topic 6 for Dr. Maduro's BIO005A class. And to jump right into it, we'll go over prokaryotic and eukaryotic cells first. So prokaryotic cells have no membrane-bound organelles. They have circular DNA, and it's packed into a structure called the nucleoid. And they're a lot smaller than eukaryotic cells. And something important is that the DNA is free in the cytoplasm, and same to the ribosomes. And so while the DNA is being transcribed into RNA, a ribosome can attach to that RNA and start translating it. So essentially in a prokaryote, you can have transcription and translation occurring at the same time. In eukaryotic cells, it gets a little bit more complex. We have a lot more membrane-bound organelles. Our DNA is stored in the nucleus as a linear structure, and our cells just are a lot more larger than a prokaryotic cell and more specialized. We also have a plasma membrane instead of a cell wall, as you see in prokaryotic bacteria, and also bacteria also have a capsule that protect them as well. Moving on, the endomembrane system is a collection of membranous structures, specifically like organelles, involved in transport within the cell. So this refers to like the nucleus, the ERs, vesicles, the Golgi, vacuoles, and lysosome. And basically, you'll see how this comes into play when we talk about protein synthesis and how it's like secreted throughout the different organelles in the cell. So the nucleus is the part of the cell that stores DNA in linear form. It's also continuous with the ER, as you can see here, so they both have a continuous membrane. Also, replication and transcription take place here. So DNA to DNA or DNA to RNA. Right. So the endoplasmic reticulum includes two types. So there's the rough ER and there's a smooth ER. What makes the rough ER rough is that it has ribosomes bound to it, which translate mRNA into proteins and pack them into like vesicles, eventually to be secreted to various locations. The smooth ER doesn't have any ribosomes bound to it. It's important for metabolism, detoxification, and ion storage, like calcium ions. And an important thing to remember is that they are both continuous with the nuclear membrane. So this is the Golgi Apparatus. It's also known as like the packaging center or like the post office of the cell. And basically, importantly, it's not continuous with the ER or nucleus membrane. It's its own entity. And ribosomes do not interact with it. So once the protein is synthesized in the ER, in the vesicle, that vesicle comes towards the cis end of the Golgi, which is the receiving end. It gets packaged or modified as it travels through the Golgi, and then gets shipped out to the trans end of the Golgi in the vesicle. And the protein has certain signals on it that tell it where to go. So it can go to like the plasma membrane, it can be secreted, it can go to a lysosome and whatnot. Finally, a ribosome. So a ribosome is like a protein synthesis or translating machinery, and it basically contains two subunits, and they latch onto the mRNA when it comes out of the nucleus. And basically, if it's going to be a ribosome that needs to be bound to the ER, the mRNA will contain a signal sequence and is going to tell the ribosome, hey, I need to go be bound to the ER because I'm a secretory protein. If not, this mRNA will just be simply translated in the cytosol and get released in the cytosol. And these proteins are mostly targeted for like peroxisomes or like the nucleus or mitochondria. And so this is the tRNA. It binds to the ribosome and it translates into a protein over here. All right. This is probably the most important slide of the video, and it basically summarizes the two types of protein transport. So you can be translated on a free ribosome, or you can be translated on a bound ribosome. All right, so I'll go through the free ribosome one first because it's a lot more simpler. So basically, your mRNA comes from your nucleus and it enters the cytoplasm. In the cytoplasm, it would bind to a free ribosome and be translated into a protein in the cytosol. And that protein can be targeted for the mitochondria, peroxisome, chloroplast, if it's a plant cell, or the nucleus. However, if it's going to be secreted or go into a plasma membrane or a lysosome, it's going to have to follow a different path. So basically, the other path is an mRNA is transcribed. It comes out of the nucleus into the cytosol. It binds to a free ribosome, and as soon as that ribosome translates a signal sequence, that sequence would say, hey, I need to be bound to the endoplasmic reticulum because I'm a secretory protein. So once it gets translated on a free ribosome, the signal sequence part, the whole entire ribosome, including that sequence, will go to the ER. ER is where it would finish translating that protein. It would put it in a vesicle, and that vesicle gets transported into the Golgi apparatus. So this is the cis end right here. It travels to the Golgi, the protein gets modified, or anything that needs to happen to it gets packaged, and the Golgi is responsible for shipping it out to the outside of the cell. The plasma membrane, if it's like an integral protein, or if it's an enzyme, it'll go to the lysosome, if it's like a digestive enzyme, or just other endomembrane structures. And so importantly, we talked about insulin in class. So insulin is a hormone, so it travels through your blood, right? However, it's manufactured as a protein because insulin is a protein. And since it's going to leave your cell, it's going to have to go through the co-translational transport route. So this route right here. So the mRNA of insulin gets transcribed. It enters the cytosol. It starts to be translated on the free ribosome. It has a signal sequence saying, hey, I'm a secretory protein. So that will be taken to the ER. Once insulin is translated as a protein, it gets packaged and transported in the Golgi, which ships it out to the exterior of the cell through a vesicle. Then once it's in the blood, insulin can travel down through the stream and bind to different receptors on various cells and import glucose for glucose breakdown. So lysosomes, as we saw in the slide before, are part of the co-translational transport, meaning the proteins made for the lysosome are secreted through the ER and Golgi. And so what happens is these enzymes, which are proteins, these enzymes get dropped off through that bound ribosome pathway into a lysosome, right? So they're digestive enzymes. And the function of a lysosome is to digest food or break down materials from the cell. So basically, if there's food like a macromolecule, these enzymes are responsible for digesting it. And a key thing to remember is that lysosomes work under low pHs, so they're acidic. And these enzymes digest the food into different particles that can be used for the cell. Or if there's cell machinery that needs to be broken down, it's called autophagy. So basically the lysosome will basically engulf or like come and help destroy it into particles so it's not harmful for the cell. If you have a mutation in the protein, you'll have like an abnormal enzyme that wouldn't work properly basically, and this could prevent cell digestion. And this leads to diseases like the one Dr. Maduro talked about, which was Tay-Sachs disease, which basically causes an accumulation of lipids in your cells and eventually causes death. So vacuoles carry a couple different functions depending on what cell we're talking about, but they mostly store water and ions. Plant vacuoles, plant cells have usually a large central vacuole, contains cell sap, but also the plant vacuoles can also function as like digestive vacuoles like lysosomes. And animal cells have various small little vacuoles going around everywhere. Also, we got peroxisomes. So this was part of the free ribosome pathway, if you remember. And basically, an important thing to remember is that it's not part of the endomembrane system. Even though it has a plasma membrane, it's not part of the endomembrane system. And it contains enzymes to break down certain molecules, and it generates hydrogen peroxide, essentially, where it gets its name peroxisome from. And that can be used to detoxify alcohols or fatty acids. Finally, we got an important structure that almost all eukaryotes have, and it's called the mitochondria, also known as like the powerhouse of the cell. You can see that it has a lot of surface area because it has a lot of folds, right? They're called the cristae. And an important thing that will help us understand the Endosymbion Theory is that the mitochondria contains its own DNA and ribosomes. It can essentially perform translation, transcription on its own. And that's important for that endosymbiont theory. And most importantly, ATP synthesis occurs here. And it's basically a very specialized organelle, and it can carry various different functions like cellular respiration as well. Similarly, we have a chloroplast, which is found in plant cells or algae. And its main function is it's responsible for like photosynthesis. And it also has its own DNA and ribosomes, which is important for the endosymbiont theory. And a key thing to remember between mitochondria and chloroplasts is that their DNA is actually going to be circular instead of like the linear DNA in the nucleus. So yeah. So the endosymbiont theory kind of predicts how mitochondria and chloroplasts came into our cell system. So basically, we talked about earlier, mitochondria and chloroplasts have their own DNA, right? They have circular DNA and they have their own ribosome. So they're pretty much like bacteria, right? And they're able to perform their own translation, transcription, and replication. So essentially, they were probably able to live on their own at one point. And so there used to be an ancestral eukaryote, as we see here, without any mitochondria or chloroplast. And there probably was a proteobacterium, which is essentially what we call mitochondria today. However, it can stay on its own, right, because it has its own DNA. And what they say is that this eukaryote engulfed the proteobacterium. However, it did not digest it, right? It lived together in symbiosis, which is where the symbiosis and endosymbiont theory comes from. And it sort of just became part of the cell, right? Essentially, the same thing happened with chloroplasts. There was sort of a bacteria called the cyanobacteria, which is pretty much a chloroplast, however, it lived on its own. And it also got engulfed into the cell. And it eventually became an organelle that we use. And this they kind of can prove because we have seen the circular DNA and ribosome machinery in both these structures. Key thing that this proves also is that they're not part of the endomembrane system as well. So the cytoskeleton is mostly for like cell support. We see it exists in the cytoplasm, but the cytoplasm, if there wasn't a cytoskeleton, then all the organelles and things would just be moving around the cell, floating on its own. However, that's not the case. We know that movement in the cell is very controlled, right? So three fibers essentially that are important for the cytoskeleton are the microtubules, microfilaments, and intermediate filaments. So we'll start off with microtubules. An important thing to remember is that they're all proteins. They're all made up of different proteins, all right? So microtubules are the largest of the three, and they're made up of a protein called tubulin. And microtubules are important in like cell motility. So like the flagella of a cell that allows it to swim, well, that's made of tubulin, right? It's a microtubule. Also, it's important for chromosome movement and cell division. So when like chromosomes align in the middle or like separate during like anaphase, the microtubules are responsible for pulling the chromosomes around. And it's also important for organelle movement in the cell. Microfilaments are usually made up of a protein called actin, and they're the smallest of the three. And they're important in cell shape, and they also come into play during cell division in something called the cleavage furrow. So a quick explanation of what the cleavage furrow is. So say this is mitosis going on over here, where we have one cell becoming two. So when two cells are ready to go, however, that cell hasn't completely divided yet, it kind of starts forming this furrow where it's gonna kind of pinch, right, into two cells. And this is what actin or microfilaments is responsible for, creating this cleavage furrow over here. Finally, we got intermediate filaments. And they're called intermediate filaments because they're in between the size of microtubules and microfilaments. So they're in between. And basically, they're made of a protein called keratin. And they're important in like cell shape, and most importantly, anchoring organelles and the nucleus. So that's why we just don't see organelles just floating around everywhere. Intermediate filaments kind of anchor them down. So finally, we have something called the extracellular matrix. And this exists on the outside of the cell. So it's actually anchored to the plasma membrane on the outside, as we see here. It's held together by proteoglycan complexes, which are made up of carbs and proteins. And its important function is that like it helps between cell-to-cell interactions or even like cell signaling. So it's pretty essential to a cell. Finally, I'd like to end with like a little analogy that I created. Basically, a cell kind of functions like a city. So what I'm trying to get at is like a nucleus. It's sort of like the city hall, right? That's where everything is stored. The basically the control center of the city or the cell, right? The DNA is contained in the nucleus, so it's like a file in the filing cabinet, and it contains all the blueprints for whatever needs to happen in the cell, right? And you can think of the ribosome as like the construction company that takes those blueprints and builds things, right? Like proteins. And they can build things like anywhere, right? And they can even go to the ER. So the ER is a little controversial, but I just named it the highway because it has like vesicular transport system. And then there's the Golgi Apparatus, which you can think of as like the post office, since it's responsible for like packaging and shipping. Vesicles are important for imports and exports. So we've seen it in endocytosis and exocytosis, or even when proteins need to be secreted out of the cell. So that happens through vesicles, right? Mitochondria is like the power plant or like the energy provider in the cell. So it provides ATP, right? Chloroplast you can think of in plant cells as like the solar energy plant because it converts light energy to like sugar. The lysosome is like the city waste disposal because it basically digests different things and autophagy and such. Finally, the vacuole carries very different functions, but you can think of it as like a warehouse or like a water tank, basically used for storage. The cytoskeleton, as we just talked about, is like infrastructure support. And if you have something like the microtubule, it's more of like the roads that things can travel on. And finally, we got the cell membrane, which you can think about as like the border security. And do you remember the plasma membrane is very like specific, right? So only some things can be entered, and only some things can leave the cell as well. So yeah, this is just like a little overview. Hopefully it helps you remember some organelles. But other than that, that's it for today's video, and good luck this week.